My name is Damir Klepac from the University of Rijeka, Croatia. First, I have to apologize for wearing the mask because we have very severe restrictions here regarding the mask, and I could be punished if I don't have it. So please forgive me.
Sure.
And I am very grateful for invitation to give this talk, especially to my dear friend and colleague Sergei Filipov. And I want to thank all the other organizers who invited me. And today, the title of my talk will be the nitroxide radical containing nanoparticles with controlled radical release that we did together with my friend Sergei. So, as you know, the trends in overall cancer mortality rates in the United States from 1930 to 2017 show that until 1990, the overall cancer mortality rate was increasing, and after 1990, the trend is steadily decreasing up to today. Many people say it's because of less smoking in general population, but probably part of it must be also because of development of novel cancer therapeutics. So today, there are several conventional drugs that are used for the treatment of tumors, and some of them, as you know, are doxorubicin, paclitaxel, and docetaxel. However, in the recent years, there are some groups that try to use nitroxide radicals as potential anti-cancer drugs. The nitroxide radicals, such as this one, tempone, are small molecules that have unpaired electron between the nitrogen and oxygen. And those radicals have been shown as the scavengers of reactive oxygen species that can cause oxidative stress and cell damage. And also, it was shown that these radicals can induce apoptosis in cancer cells and suppress tumor growth. So basically, they can be used as another potential anti-cancer drugs. However, those nitroxide radicals have several disadvantages. One of them is that they have limited efficiency. They are distributed non-selectively, so they have equal concentration in cancerous tissue and the normal tissue. And because of their low molecular masses, they are very rapidly excreted from the human body. These are all disadvantages of nitroxide radicals that prevent them to be successfully used as anti-cancer drugs. However, we can use maybe controlled drug delivery in order to overcome these disadvantages. By using controlled drug delivery, the drug can be transferred directly to the site of action. The drug can be protected from rapid degradation, and the nanoparticles can be used as drug delivery carriers in this drug delivery method. So in our group, we investigate polymer nanoparticles, and this talk will be mostly based on polymer nanoparticles. These are known as versatile carriers for the delivery of a wide range of drugs. They provide control over the drug biodistribution and pharmacokinetics. And this is a schematical picture of this such nanoparticle that contain hydrophobic core and hydrophilic corona. And the drug can be either encapsulated in the hydrophobic core or covalently bound to the hydrophilic corona. These nanoparticles improve stability of the drug during blood circulation. It is already known for several drug formulations. So our research goals was first to prepare nitroxide radical containing nanoparticles based on the N2-hydroxypropyl methacrylamide, and to investigate the release of nitroxide radicals from these nanoparticles at physiological pH and at pH 5, which corresponds to the pH of endosomal compartments of cancer cells. So first, we synthesized polymer conjugate based on poly-HPMA and cholesterol by Raft polymerization of hydroxypropyl methacrylamide, spacer, and hydrophobic cholesterol moiety, and the polymer precursor was first synthesized. In the next stage, dithiobenzoate end group was removed, and tempo radical was covalently bound to a polymer precursor to give final polymer conjugate. Another diblock copolymer was created by ring opening polymerization of caprolactone, and free polymer was created in the first step. In the next step, polycaprolactone macromolecular chain transfer agent was created by carbodiimide chemistry. And in the next step, by rough polymerization, final diblock copolymer containing from PCl block, polycaprolactone block, and hydroxypropyl methylamide block was synthesized. And this diblock copolymer was in the last reaction oxidized to afford stable nitroxide radical covalently bound to the hydrophobic PCl block. In this way, two diblock copolymers were created, or polymer conjugates. First one containing PHPMA as a hydrophilic block and cholesterol as a hydrophobic moiety, and the other one, and in this diblock, in this polymer conjugate, nitroxide radical was covalently bound to the HPMA backbone by shorter and longer spacers. The other type of diblock copolymer, as we already said, is based on poly-HPMA and polycaprolactone, and in this case, the hydrophobic radical is covalently bound to the hydrophobic block. These polymers were nanoprecipitated to afford nanoparticles, and the nanoparticles were later dialyzed in order to remove the organic solvent and put them in the phosphate buffer saline. These nanoparticles, after nanoprecipitation, we had two types of nanoparticles. First one, that have radicals covalently bound to the hydrophilic corona, and the other type of nanoparticle that contain radicals in the dense hydrophobic core. And those two nanoparticles were then characterized by cryotransmission electron microscopy, and indeed nanoparticles were created. And by dynamic light scattering, it was shown that the hydrodynamic radius of such nanoparticles is between 40 and 50 nanometers. After this basic characterization, the nanoparticles were thoroughly characterized with electron paramagnetic resonance spectroscopy, which is the main group in our laboratory, main technique in our laboratory. ESR spectrum has this let's say, consists of three lines, and such ESR spectrum behaves differently with different mobility of the nitroxide radical. So we can distinguish two basic types of ESR spectra. First one, narrow spectra, which is characteristic for fast motions of nitroxide radical, and another type of ESR spectra that is characteristic for slow motions of nitroxide radical. Also, the shape of ESR spectra can also be simulated in order to get so-called rotational correlation time, and the rotational correlation time is the typical time which takes for a radical to rotate 1 radian. In the case of fast motions, the rotational correlation time is in the range of 10 to minus 11 to 10 to minus 9 seconds. And in the case of slow motions, correlation times is between 10 to minus 8 to 10 to minus 7 seconds. So the rotational correlation time can be a sensitive parameter that can characterize the mobility of nitroxide radicals attached to the polymers. Here we can see the ESR spectra of free tampon radical. When the radical is free, the spectrum is very narrow and the lines have almost equal intensities, and the rotational correlation time is very slow. However, after attachment of this nitroxide radical to the HPMA polymer backbone, the high field line has lower intensity and the rotational correlation time increases because of the restriction of mobility of this nitroxide radical. By shortening the spacer length in the other type of nanoparticle, the shape of ESR spectra becomes broadened and the rotational correlation time even increases further. So by shortening the spacer length, the mobility of radical is even more restricted. In the case of of nanoparticles based on polycaprolactone and PHPMA, there is a big difference in the shape of ESR spectra before and after micellization, because the radicals are now placed in the dense hydrophobic core, where their mobility is very severely restricted. So first, we wanted to show the stability of nanoparticles in sodium dodecyl sulfate, which is known as strong detergent that can destroy the structure of nanoparticles. And indeed, before adding sodium dodecyl sulfate, the nanoparticles are clearly present. And after adding sodium dodecyl sulfate, the nanoparticle structure was destroyed. When we look at the ESR spectra before and after adding sodium dodecyl sulfate, it can clearly be seen that the slow motion spectra, which has 3.8 nanoseconds correlation time, become fast motion spectra with much faster mobility. This is indeed expected because nitroxide radicals, which were previously in the dense hydrophobic core, after adding SDS, suddenly become exposed to the solution. And in the solution, they can move much more freely than before when they were restricted. However, in the case of nanoparticle where the radicals are connected to the corona, there is no change in the shape of ESR spectra, because regardless of the integrity of nanoparticle, these nitroxide radicals can move much more freely than in this case. So here we just wanted to show that the ESR is indeed sensitive. ESR spectra are indeed sensitive to the motional changes in the structure of nanoparticles. So once we establish that, we use this property of ESR to try to see how will be the interaction of nanoparticles with real blood environment. So we mix these nanoparticles with various proteins present in the blood, and finally with human plasma. Here there are results presented, results of ESR, and we can see that for both types of nanoparticles, regardless of protein used, and regardless if you put the nanoparticles in plasma, there was no change in ESR spectra. These were quite surprising, because usually nanoparticles, when they're injected in the blood, they encounter a lot of proteins which interact with them, and these proteins are usually deposited on the surface of such nanoparticles. However, if that would be the case, here we would see that the correlation time and the ESR spectra becomes much more broadened because of the restricted mobility. However, there was no change of ESR spectra, of the shape of ESR spectra, and also in the corresponding rotational correlation time. The same, it was the case in the other type of nanoparticle, which showed that none of these proteins can penetrate the HPMA corona and influence the radicals in the nanoparticle core. Here we can conclude that nanoparticles based on HPMA, when they are injected into the blood, have no strong interaction between proteins and nanoparticles, and no hard and soft corona can be formed around nanoparticles, which is quite surprising, because most other nanoparticles usually form this at least soft corona around nanoparticles. So this was our first paper, which we published with Sergey, and now I will show you new results, which will be published very soon, about the same type of radical containing nanoparticles. We prepared two types of nanoparticles, again based on poly-HPMA and cholesterol. In one type of nanoparticle, the nitroxide radical was bound to the HPMA block with hydrogen bond, and in another type of nanoparticle, the nitroxide radical was covalently attached by peptide bond. So the only difference is the type of bond by which the nitroxide radical is attached to the HPMA chain. Nanoparticles were created with the nanoprecipitation method, and two types of nanoparticles were created, ones with the hydrogen bond between the nanoparticle and radical, and another one with peptide bond between the nanoparticle and radical. These nanoparticles were incubated for various amounts of time at different pH values, from 7.4 to 5, to see how the pH of the medium will influence the release of these radicals from nanoparticles at different pH levels. So here we can first see the ESR spectra of free tampon radical and the ESR spectra after attachment of this radical to the nanoparticles. Clearly, the spectrum becomes more broadened and this high field line becomes lower, and also rotational correlation time increases, which means that the radical were successfully bound to the nanoparticle. After 24 hours of incubation at various pH values, we can see that already at pH 6.8 there is some release of radicals, but at pH 6 and especially at pH 5, the spectrum is completely the same as the one of unattached radical, which is this one. That means that at pH 5 and even most at pH 6, after 24 hours, the radicals are indeed released into the solution. And in the case of nanoparticles that contain radicals bound with peptide bond, there is no change in the shape of ESR spectra or in the change in the correlation times, because it was shown that the peptide bond is indeed stable even after 24 hours at all these pH values. Then here we can see the ESR spectra after different incubation times at the lowest pH that corresponds to the cancerous cells. And already after 3 hours, almost all the radicals are released from the nanoparticles. And after 24 hours, indeed, the spectrum is the same as for the completely free nitroxide radical. So once again, at pH 5, the radicals are very easily released almost after, even after 3 hours. Again, for comparison, another type of radical of nanoparticles with a peptide bond, there was no change at pH 5 at any incubation time. So putting it all together, here we can see that for nanoparticles with hydrogen bond, at pH 7.4, there is no significant release of radicals from nanoparticle. By decreasing the pH level, the release of radicals is stronger, and at pH 5, already after 3 hours, almost all of the radicals are released from these nanoparticles. Again, for comparison, the nanoparticles with peptide bond, there is no significant change of correlation times, which means that most of radicals are still connected to the polymer backbone. So finally, we can conclude that we develop nanoparticles based on PHPMA and cholesterol that contain nitroxide radicals bound to the polymer carrier by a pH-sensitive hydrogen bond. These nanoparticles show that almost no interaction with blood proteins and no hard corona was detected around the nanoparticles. And the nitroxide radicals are quickly released from nanoparticles at pH 5, which corresponds to the pH of cancerous cells, while there was no significant release of radicals at physiological pH. So these nanoparticles could be potentially used as nitroxide radical carriers to prolong their circulation in the blood and increase the accumulation of radicals in tumor tissue. And finally, I would like to thank my colleagues who contributed to this work, especially the best one, Sergey, and other colleagues from the Institute of Macromolecular Chemistry, such as Peter Hitil, Svetlana Petrova, Hanna Koskova, and my master student, Martina Dragovic, who recently joined the Institute as a UNESCO student. And also Semi Kereich and Lubomir Kovacic from the First Faculty of Medicine at the Charles University in Prague. And finally, I would like to thank you for your attention, and I invite everybody to Croatia, into Dubrovnik or any other place. So thank you very much.
Okay, thank you very much. There's a couple of questions in the chat. First question is from Xiaoning Shang. Thank you for your talk. 2 questions here. The first one, why the nitroxide radicals are distributed in the hydrophilic corona of poly-HPMA cholesterol, while distributed in the hydrophobic core of poly-HPMA PCl instead?
I didn't get the question really. Can you repeat, please?
So why the nitroxide radicals are distributed in the hydrophilic corona of poly-HPMA cholesterol,
Okay.
while distributed in the hydrophobic core of poly-HPMA PCl?
But those are two different nanoparticles. Here, in this case, where the radical is bound to the hydrophilic corona, it was a statistical, how to say, statistical. That I don't know how to describe this. There was no some particular way in which this was distributed. It was distributed randomly. And here in the hydrophobic corona, it was distributed at the end of these chains. It was synthesized like that.
Okay.
Yes. So.
Okay, thank you. And the second question from her, what about the drug loading for these two types of polymer matrices?
Yes, any drug can be. So if we see the structure of these divalent copolymers, we can see that whatever drug has these groups which can be connected in this place can be easily loaded to these nanoparticles. So by using this synthetic procedure, after removing of this dithiobenzoate group, whatever can be covalently attached to this group can also be loaded to the nanoparticle. So this is how it's usually done.
Okay, thank you. And next question is from Felix Plumper. Can you derive the rotational diffusion coefficient of whole micelles with tempo in frozen core from ECR data?
Yes, I can, because once we measured the ESR spectrum, I didn't show this in detail. Once when the ESR spectrum is measured, there is a mathematical procedure and a special software that is created for the simulation of such spectra. And let me see if I have some. You see these dashed lines. Those are simulated spectra, which means that from the shape and usage of this mathematical procedure, it is possible to get the mobility of any part of the molecule in the vicinity of attached nitroxide radical. Even if there are different mobilities in the same nanoparticles, we can have different components in the ESR spectra, one with lower mobility, another one with higher mobility, and by analyzing these components, it is possible to see the mobility of various parts of these nanoparticles.
Okay, thank you. And another question from Felix. Did you check the electroactivity of the tempo micelles?
No. This could be a part of the next investigation, and also the zeta potential can be also the part of next investigation, which will be which we'll probably do in several months. Yes.
Okay.
Mm-hmm.
There's another question from Andrei Lashevsky. Where do you see the particular advantage of cholesterol as hydrophobic unit instead of using standard units as, for example, oleal chains?
Yes, the group in the Institute of Macromolecular Chemistry are pioneers in developing such nanoparticles. And since they are new and have these stealth properties, and which means that they don't much interact with proteins, we thought that they have much more advantages than another nanoparticles that were previously used that form protein corona. Those can be used as stealth nanoparticles, and since they have these unique properties, we just want to investigate them as much as possible. So this was just an opportunity which was shown because of these special properties. If some people have some idea for another type of nanoparticles, I will be very happy to make some collaboration and measure also these properties. I'm always happy to get new samples with attached nitroxide radicals, so we can play with ESR spectra and do different investigations. So why not another nanoparticle would be also good for this type of investigation.